again, everyone, and welcome to another NIRx webinar. It's a great pleasure to have you with us today. I'm especially happy to welcome our guest speaker, Dr. Georg Kranz, who is an associate professor at the Department of Rehabilitation Sciences at the Hong Kong Polytechnic University. We are really excited about today's talk, and we've been looking for this one for quite some time. The title of his presentation is From Coil to Circuit, Using Imaging to Map and Optimize TMS Effects. Today, Dr. Kranz will introduce the fundamentals of TMS and also discuss both the challenges and advantages of combining it together with fNIRS. And this is actually the first part of our short TMS fNIRS series. Tomorrow we will continue with part 2, where PhD students Adam and Jimmy from Dr. Kranz's lab will take us behind the scenes, and they will talk more about the practical challenges they faced running concurrent TMS fNIRS studies, share their solutions, and walk us through basic preprocessing and data analysis of TMS fNIRS signals. So definitely join us again tomorrow. It's going to be great. And before we continue, I would like to give a quick shout out to Kami, our wonderful marketing and events coordinator, who is here helping me today to moderate this session. And if this is your first time joining our NIRx webinar, welcome. And let me give you a very quick intro to our company. For more than 20 years, we've been designing and manufacturing state-of-art fNIRS systems, which are made in Berlin and distributed all over the world, including Hong Kong, as you can notice today. Our mission is simple: to empower researchers. We have an amazing team of scientific consultants supporting scientists at every stage of their projects. And these webinars are part of our effort to share the latest and most exciting developments in fNIRS, like today's work at the intersection of TMS and optical imaging. You can always connect with us across different platforms, and if you have any questions at any point, just send us an email at consulting@nirx.net. You can also subscribe to our newsletter. Just scan this QR code on your screen or visit nirx.net/newsletters. That's where you'll get updates on our newest product features, software releases, and highlights of innovative research from the community. And don't worry, we promise not to spam your inboxes. We usually send a few emails per year. And before we begin, just a few quick housekeeping notes. Everyone on this call is muted, but of course we love questions, so please type them anytime into the Zoom chat, and we will address them during the Q&A at the end of this talk. This session is being recorded, and you will be able to find the video soon on our website and YouTube channel. And of course, if you would like to follow up afterwards, you can always reach us again at consulting or support@nirx.net. All right, without further delay, let me introduce Dr. Georg Kranz. Dr. Kranz is an Austrian neuroscientist and currently working as an associate professor at the Department of Rehabilitation Sciences at the Hong Kong Polytechnic University. Together with his team, he explores how non-invasive brain stimulation therapies can help us better understand and treat neuropsychiatric disorders. Over the years, Georg Kranz has published more than 120 scientific papers in top-tier journals such as the Lancet Psychiatry, PNAS, and JAMA Psychiatry. And Dr. Kranz, we are very excited to have you with us today, and the virtual mic is yours now.
Okay, thank you very much. Now I will share my screen. Okay, so here we go. Do you see the screen? Yes. Good. Okay, good. Yeah, I'm in Hong Kong, and it is evening, so it's dark outside. But for some of you, it is daytime, and I heard maybe there is even people from the US where it is in the middle of the night. Who knows? If you join, thank you very much for joining, and thank you for inviting me, for giving this this webinar, this talk today. Tomorrow, Adam and Jimmy will talk about you know their studies that I will present just as a quick overview today, and they will so all the technical things. If you have really in-depth issues or things you want to ask about the combination of TMS and fNIRs, please ask them. I will just tell you the reason why we do this, why we combine TMS with fNIRs, and yeah, what kind of the background. Okay, this is my conflict of interest disclosure slide, and originally I'm from Vienna, from the Department of Psychiatry and Psychotherapy, so my interest is in psychiatric disorders, and since coming to Hong Kong, I focused on brain stimulation in psychiatric disorders, in depression mostly, and in combination with imaging. And this is my team here that you can see some of them. And yeah, to be very honest with you, when I came to Hong Kong, there was no MRI machine in my university. They bought it a year later, and there was no positron emission tomography, which is also, an imaging technique that I have been using in Vienna. So for these reasons, I looked into the application of fNIRs. Sorry for being honest, but this was the first reason. And then I realized actually fNIRs is quite cool to be combined with TMS. Okay, so this is the agenda. First, I will give an introduction to non-invasive brain stimulation and to TMS in specific. And then I will talk about our research combining TMS with neuroimaging with fNIRs. And I will start with the introduction to TMS and to non-invasive brain stimulation, assuming that you have only little knowledge. But if you have a lot of knowledge, then ignore. Then ignore it. Have a coffee and come later. Okay. So brain stimulation, as you may know, is there's different kinds of brain stimulation, and these techniques are used in psychiatry more and more. Some of course have been used for already decades, such as electroconvulsive therapy, but these non-invasive techniques are being more and more investigated recently, and that's why some psychiatrists have suggested that we should actually have a separate subspecialty in psychiatry called, for example, interventional psychiatry. Some propose a different name. And this interventional psychiatry subspecialty utilizes neurotechnologies to identify brain circuitries, dysfunctional brain circuitries that proposed to be underlying in psychiatric disorders, and then applies neuromodulation or brain stimulation techniques to modulate, to counteract this dysfunctional circuitries or activity. Yeah, this was published by Nolan Williams in 2014. Nolan Williams, maybe you know, or some of you may know, passed away last week, very tragically. Okay, so brain stimulation includes non-invasive and invasive forms. Non-invasive includes, for example, TMS, transcranial magnetic stimulation, transcranial electrical stimulation, and the most famous form is TDCS, transcranial direct current stimulation, but there is also other forms. And then we have focused ultrasound, very attractive, very sexy these days. Everybody tries to use focused ultrasound, does research with FUS, also in combination with MRI, for example. And then we have photobiomodulation, and we have electroconvulsive therapy, and so magnetic seizure therapy, the convulsive techniques that are, although you intentionally trigger a seizure, nonetheless, they are non-invasive because you are not implanting anything, right? You're not breaching the integument, the skin. And then we have invasive techniques such as deep brain stimulation, such as vagus nerve stimulation, although traditionally vagus nerve stimulation. There is nowadays also non-invasive forms. Okay, I will focus on TMS today. TMS, as the name says, transcranial magnetic stimulation, is a stimulation that goes through the intact cranial bone, is magnetic in nature, and stimulates neurons. So as compared to TDCS, for example, or transcranial electrical stimulation, where you only modulate the excitability. With TMS, you can really induce action potentials. And what you need basically is a stimulator and a coil. And there is different coil types, but this is basically it. And the theory behind it, or the law behind it, is electromagnetic induction. So in a coil, if there is a current going through a coil, this induces a magnetic field. And this magnetic field then induces a secondary current in the brain. This secondary current then induces action potentials if the intensity is high enough, right? And this is oriented perpendicular to the magnetic field. The electrical field is the induced electrical field is then in opposite direction and proportional to the magnetic field. And it depends on how strong you turn on the machine. The magnetic field that is induced in a very short period of time can be up to 4 Tesla. So this is a very high magnetic field. If you go 100% machine output, you can induce a very high short magnetic field. Now, if we stimulate on the motor cortex, if we perform a TMS, a single pulse on the motor cortex, we can have a direct readout. We can measure the stimulation by performing electromyography, by looking at motor evoked potentials. Can you see my cursor? I assume so. Elena, can you see my cursor?
Yes, we can.
I only see you now. You are nodding. I guess I'm sure you must see my cursor.
Yes, we do see.
Good. So you have the TMS pulse, and then you see a motor evoked potential in the muscle that you basically stimulate, or you stimulate the receptive field of the motor cortex of this muscle, and then you have a direct readout. And this is nice to see, but it is also relevant for treatment, because this is a way to standardize intensity. So for TDCS, no, not to standardize, to individualize intensity, actually. For TDCS, you do not individualize intensity. You stimulate with 2 milliampere or whatever. But for TMS, you apply an intensity that is individualized to the motor evoked potential that you measure before you started treatment. Because what you would do is you would determine the so-called motor threshold that is defined as a certain amplitude, or you can also do it visually. If you stimulate on the motor cortex of this hand area of the specific muscle that you want to look at, yeah, and you stimulate, let's say, with a very low intensity, and then you go up and higher and higher and higher. At a certain period, at a certain level of intensity, you will see a twitch. You will see a twitch. You either see a MEP or you see visually a twitch in the muscle. And this motor threshold is individually different. So for some people, it is at a very low intensity. For other people, it's very high at a higher intensity. It's individual, and it depends on the gyrification and the distance from the scalp to the cortex and so on. Yeah, and several other factors. But this is what we basically use to individualize the treatment, that the stimulation intensity that is then applied on other brain regions. Because for psychiatric disorders, for example, we are interested in more frontal regions. Yeah? So we assume that this works, that the intensity is just enough to reach the motor cortex also applies for the frontal cortex. Yeah. So we assume the distance is the same, for example, or the excitability of the frontal cortex is the same. Because then in the end, we apply a treatment with respect to this motor threshold. For example, you will read papers that say we stimulated at 100% of the resting motor threshold, or 120%. Which is the FDA-approved protocol for depression, for example. And this is a link, but yeah, maybe I click into it, and then you have a quick view. Sorry, this is a Chinese advert.
自信就係素顏依然晶瑩剔透 ，SK2 神仙水 ，Pitera 三大功效。我嘅
Maybe this takes too long. Yeah, sorry. It takes too long. You can click on your link. You can copy the link later and look at it. What I think this video shows you just the muscle twitch when you stimulate the motor cortex. So let's continue. Okay. Good. Now you can apply TMS as a single pulse or paired pulse protocols, or you can apply it repetitively. And if you apply it at a single pulse on the motor cortex, then you have a direct readout. You can see the online effect, right, of the stimulation. You can also apply repetitively and see the online effect, as in you can see that the stimulation disturbs ongoing brain activity and ongoing brain processes. For example, if you stimulate the Broca area while you want to speak, while you produce language. And this is also a link. And if you want, let's click into it. Let's see if there is a long advert. No, directly goes to the video. So let's watch this.
I'm Roger Highfield. I'm the science editor of the Daily Telegraph. Now, words mean everything to me, but now I'm going to have the speech area of my brain turned off with a giant magnet. Humpty Dumpty sat on a wall. Humpty Dumpty had a great fall. All the king's horses and all the king's men couldn't put Humpty together again. Humpty Dumpty sat on a wall. Humpty Dumpty had a great fall. All the king's horses and all the king's men couldn't put Humpty together again.
Good. Okay. Now, on the other hand, in the early 90s, people realized that there is after effects of a repetitive stimulation. So if you stimulate repetitively, for example, on the motor cortex, then we can see after the stimulation that the MEP, the motor evoked potential amplitude that we can measure before and after with a single pulse, for example, changes. And this was an indication for the fact that TMS can modulate cortical excitability after we stop the stimulation. Yeah? And this hints towards neuroplastic changes. And what people realized is that these plastic changes or the modulation of cortical excitability seems to depend on the frequency of the stimulation. So if we stimulate with 1 Hertz every second, then it seems to inhibit the cortex. And if we stimulate with a higher frequency, 5 Hertz or above, 10 Hertz, for example, is a common protocol, then we can facilitate, we can increase cortical excitability. And suddenly, researchers had a tool in their hand to focally modulate brain regions that they already had an idea about being altered in psychiatric disorders. For example, early imaging studies in the 80s, 90s showed that depression might be associated with not enough activity in the left DLPFC, left dorsolateral prefrontal cortex. Or there is indication, there is evidence that tinnitus might be associated with too much activity in auditory, right, cortices. That's the reason why the cortex produces this annoying sound, although there is no sound in the real world. And this was very exciting. But let me go back to the early beginnings. So, and I like this because I come from Vienna, and to find out, yeah, everybody, all the TMS researchers have a different way of introducing TMS in the history of TMS. But I like the fact that the first patent for TMS for depression was actually filed in Vienna by 2 psychiatrists. They got it totally wrong. Yeah, they thought the vibration may have an effect. But they patented it, and then the first pilot trials, so to speak, yeah, were also done. Were not done in Vienna, but by my old boss, who was old boss in Department of Psychiatry in Vienna. Yeah? He was at that time, he was in Germany, but this was really just a trial and error. They tried to apply TMS. Also on CZ, on the vortex, which is nowadays we know that this does not work. But the first pilot trials were done by my old boss. However, as I told you before, these early imaging studies that showed that there is probably something wrong with the left, especially the left dorsolateral prefrontal cortex, that an early PET study, FDG PET study, showed that there is probably reduced metabolism in the left DLPFC. Yeah? These early imaging studies led to an idea, a proposed model of depression that poses that there is not enough cognitive control that controls limbic hyperactivity. So there is a limbic hyperactivity and a prefrontal lack of this control, lack of dorsolateral prefrontal cortex activity. And therefore, just a few months later, a first hypothesis-driven study was published in America. So they used high-frequency stimulation that is proposed to be activating or excitatory over the left dorsolateral prefrontal cortex every day, a treatment for several days and later several weeks. And this was really, you could say TMS is one of the few, things in psychiatry that were not purely based on a luck, yeah, that were hypothesis driven. And over so this was the beginning. And this idea that there is something wrong, that there is a hypoactivity or hyperactivity, and in order to rectify that, we use either high frequency or low frequency TMS or excitatory and inhibitory TMS. This idea prevails until today, and this is just the huge increase in number of publications over the years, but the same idea prevails, and for today we know for sure that left DLPFC rTMS is effective for depression. Has a level A evidence for efficacy, and there is FDA approvals. And for left DLPFC is really has the highest evidence. Okay. Also, what we also realized is that over the years that the risks are much lower than originally thought. Okay, this is the next slide. So, what are the side effects? You have in most cases you have sensory discomfort, so it hurts a little bit, especially if you have never experienced it before, and but then after a few days of treatment, you will get used to it. And I almost get my TMS not for treatment but for experimental reasons every week. Yeah, I do something because I'm also doing studies on my own face, my own brain. Yeah? Adam will tell about that tomorrow, I think. And I don't even twitch anymore. Maybe the first TMS pulse, the first train, we use theta burst simulation. The train is 2 seconds only. Maybe I move a little bit, but then there is nothing. Yeah? What can also be a little bit annoying is facial nerves. So during the stimulation, your jaw may move or your eyelids, but much less likely is syncopes, and the most severe, adverse event would be an epileptic seizure, but this is very, very low. Okay. As compared to antidepressant pharmacological treatments, for example. Yeah, this is a comparison. You see all these different side effects, and rTMS is headache. Yeah, but these headache or neck stiffness, these can all be treated with painkillers very simply. The only absolute contraindication is cochlear implant or any metal implants in the brain that are within about 10 centimeters from the coil. Anything else is no problem at all. But if you look at the old guidelines, safety guidelines from, let's say, 2009, for example, they still say you shouldn't use TMS in pregnancy, and you shouldn't, and yeah. So, but now we know it is much safer than we thought at first. Okay, now the standard protocol is every day a treatment for depression, high frequency rTMS of several minutes, and you come every day for 5 days a week for at least 4 to 6 weeks, and this is effective. More recently, so 10 years ago, theta burst stimulation, and I'm mentioning that because all my studies use theta burst stimulation, all my concurrent TMS fNIRS studies. That's why you should know about that. Theta burst stimulation is a protocol that uses 50 hertz triplets repeated every 5 hertz. So it sounds like that. And this protocol was developed in 2005 or published first in 2005 and showed on the motor cortex that it induces excitability if you perform this intermittently. If you have 2 seconds, these triplets on, and then 8 seconds pause, and then 2 seconds on again, and 8 seconds pause. So this is called intermittent theta burst stimulation. If you perform that for 600 pulses, that's about 3 minutes and 10 seconds, then the effect was an increase in excitability that lasted for at least 20 minutes. after the stimulation, I think. Yes, and then there was a continuous form, CTBS, continuous theta burst stimulation, where you just continue these triplets for 40 seconds, the same amount of pulses, that led to a sustained inhibition of the motor cortex. So this was very exciting because a new protocol, stimulation protocol, TMS protocol, that is much shorter in time, only 3 minutes. But yeah, the excitatory version, the intermittent theta burst, is only 3 minutes and 10 seconds. Potentially could be used also in depression, and these are two studies that this is an early study. This is then the large clinical trial that led to the FDA approval. Because now you can see these non-inferiority trials showing that the standard long treatment was comparable in their efficacy. This is the antidepressant response over the weeks to this very short 3-minute protocol. So much higher efficiency because you can treat many more patients in one day. And so that's why this is relevant for psychiatric disorders, and that's why we use it in our studies, only ITBS and CTBS in our concurrent TMS fNIRS studies. And the fact that you can use these very shortened treatment protocols led to the idea that we could maybe stimulate more than just one time a day. And these are then called accelerated protocols, where you stimulate, for example, 5 times or even 10 times per day, such as in this SAINT protocol that was published in 2020 and then replicated in 2022. A crazy protocol, basically intermittent theta burst stimulation applied 10 times per day. At 8 o'clock in the morning they started, and then at 9, and then at 10 again, at 11 and 12 and so on, until 4. Yeah, and this had in very better than real results. Yeah. In terms of remission and response rates in patients who were highly treatment resistant. Okay. As I said before, Nolan Williams, who is the inventor of this, basically, passed away tragically last week. But this is really exciting. And although many psychiatrists don't really believe in it, and because this treatment led to these amazing results after only 1 week. 1 week, 10 sessions per day for 5 days, and you had these amazing results. And many psychiatrists don't really believe it, and there is now replications underway that try to replicate it, because you cannot exactly replicate it, since they patented their way to target the DLPFC, but I wouldn't go into detail. Anyway, I also don't really believe it. Because everything we know about depression, chronicity, where is that? You cannot treat depression in 1 week, I believe. Okay, but I want to mention that because this is really one of the highlights of TMS in recent years, these accelerated protocols. Another recent advancement was that we estimate the E field, the electrical field that is induced in the brain. We look at the electrical field because the electrical field that really reaches the brain depends on many different factors. And this is, for example, one study of my former PhD student that looked into the different components of the electrical field and which components may be more relevant for depression. I will also come to electrical field estimation later. when it's more relevant for the TMS fNIRS studies. Anyway, these are the summary of the recommendations. These recommendations are now being updated, not yet published, but these are the recommendations from 2020. And you can see TMS is recommended with different levels of evidence. Level B, for example, is probably effective. Level A means it's definitely effective. TMS is definitely effective. And what you can also see is that in many instances, we stimulate the left dorsolateral prefrontal cortex, the left DLPFC. And yeah, so for different conditions even, not only for depression. And this led us to ask, and why is that the case? Because I said before, because we have this model of depression, that there is a lack of prefrontal control, but this model can actually also apply for other psychiatric disorders, for addiction, for example, or for schizophrenia, negative symptoms of schizophrenia. So left DLPFC rTMS for schizophrenia was something that was intensively studied. Had a level B recommendation, now is level C, because there was a negative, big RCT negative trial. But the left DLPFC is really one of the common TMS targets. Okay. And so we asked recently our question, this is another former PhD student of mine, for which symptoms is left DLPFC rTMS really most effective? So we performed a cross-diagnostic meta-analysis. That means we looked at many different studies and diagnoses and combined the effects. If they measured depression or anxiety, let's say, if they measured an anxiety, clinical scale that measures anxiety, irrespective of the diagnosis, we summarized them here and looked at the relative effect size. And the same for attention and so on and so on. And what we found is that actually the anti-craving effect of left DLPFC rTMS has the largest effect size and not the antidepressive effects. So this was quite interesting. And here you see basically the same thing again. For craving, largest effect size. And yeah. So this study is the first step towards the development of evidence-based symptom domains for which therapeutic rTMS is effective. Good. And yes, and this was the first part. And it's now 7:40. I only have 20 minutes left, right? It took a long time. Sorry. Then let's quickly continue. So now I move on to the second part, combining TMS with neuroimaging, brain challenge test. Now the question is, what does left DLPFC rTMS do? Because rTMS of the left DLPFC is an example of clinical applications preceding the understanding of the mechanisms of action. For the motor cortex, we have a direct readout. For the left DLPFC, we don't know. We shoot into the dark. So that's why we think we need to combine rTMS with a neuroimaging technique to find an MEP-like measurement for the left DLPFC. For left DLPFC rTMS, or a pharmacodynamic marker of rTMS in broad terms. And how do you do that? You perform simultaneous TMS with imaging, such as with fMRI, and we do that now with fNIRS, with EEG, we also do that, and we even do the triple TMS, fNIRS, fMRI. And others do triple TMS, fMRI, EEG, for example. We haven't done that yet, but we do triple fNIRS, fMRI, because we got the Borealis system from NIRx. Good. The advantage of TMS fNIRS is that we can really have a continuous assessment of the hemodynamic response, and we don't have to do things interleaved, right? What we would have to do in TMS fMRI, for example, because of the interference, electromagnetic interference of the coil with the MRI. But for fNIRS, we don't have this interference. So first we did a meta-analysis. Adam will talk tomorrow. Yeah, he performed a meta-analysis just looking into what is known about you know, what is there a common activity, a pattern of activities in already conducted concurrent TMS, fNIRS, and fMRI studies. And interestingly, there was no real common ground of these studies. Yeah. So we started with our own studies, and we found, and this is very consistent over now 6 or 7 studies that we performed, independent samples, depressive patients, remitted from depression patients, healthy controls. We always find that during the stimulation, and as I said before, we use intermittent theta burst stimulation. We don't use the standard rTMS. We only use this ITBS. During the stimulation, HBR goes up and HBO goes down. And this is consistent over all these different studies. And the question is why? Is it a direct effect on the vasculature, or is it a brain effect? Rather, yeah, the former and not the latter. But so this was published by Adam. What we also see is that there is a huge individual variability. And of course, this has also technical reasons. So we really try to minimize the artifacts. And Adam will talk about that tomorrow, and Jimmy. But what we also try to see if there is if we can explain individual variability by looking at other things, such as did the person sleep well? When do we stimulate, in the morning or in the afternoon? Perform this concurrent measurement. Female, male, is there gender differences? Yeah, whether they drank coffee or not, which of course would make sense. Yeah, all of these things, and I will just show you a few things. For example, we found that intake of caffeine has an effect during and after stimulation, whether we stimulate in the morning or in the afternoon. The excitatory protocol that is presumably excitatory, ITBS, seems to wear off if we look at HBO. And there seems to also be a correlation with mood, daily mood. Inter-individual variability of prefrontal excitability in these studies we found that males, for example, have a much stronger change than females. This also depends on the intensity. There is also a sex difference before the stimulation, so in absolute hemoglobin concentration. We use a different machine that measures absolute hemoglobin concentration. And this is then aggravated by the stimulation. But if we stimulate with an internal, so with a task, we do not see this gender difference. But I need to be a bit quicker, maybe. So now the one question is, for example, why do we have this sex difference? Could it be scalp to cortex distance? This was proposed already by other people, that the scalp to cortex distance is different in males and females. Because the head morphology is different, and therefore the induced electrical field, the intensity that reaches the brain, might be different. Now, if we also measure HBO, and there is a cortex layer dependent blood oxygenation, then the measurement should also depend on the scalp to cortex distance, and we know that this is the case. There is also publications, not from us but from others. That head size, that all these things matter, right? Most recently, a study that also showed that hair color and so on, thickness has an effect. Anyway, scalp to cortex distance is a crucial factor for the fNIRS measurement as well as for the TMS stimulation intensity. This has been shown now for TMS for quite some time. And this is also the reason why why the FDA approved stimulation of the frontal cortex with a higher intensity than on the motor cortex. So on the motor cortex, because the motor cortex has a shorter distance than the frontal cortex, larger distance. And if we use the same intensity on the motor cortex, 100% of the motor threshold. At the frontal cortex, we would stimulate the frontal cortex less. So that's why 120% of the RMT on the motor cortex is conventionally used to ensure supra-threshold intensity. So, but going back, we wanted to we wanted to know, is there a gender difference? So we looked into our own data. And what we found was, surprisingly, that the distance, scalp to cortex distance, is actually shorter in the LDL-PFC than in the motor cortex. And this was very surprising, because we thought the literature shows LDL-PFC distance is larger than motor cortex. So what is going on? So we asked the second question: not only are there sex differences that can explain the scalp to cortex distance, but also are there ethnic differences, Chinese versus Caucasians, because the FDA approved protocols, they were all based on Western studies. And what we did was then we estimated the E field. I will just skip this quickly, and let's go directly into the results. So we found that there is indeed a difference in the scalp to cortex distance in white and in Han Chinese people. And this is reflected then also in the electrical field that reaches the brain. We submitted this. I hope it will be published soon. So here you can see the scalp to cortex distance was the primary predictor for the TMS-induced E field. Of course, different data sets, so Chinese, Western, and even African American, we had a data set. And then we asked, could this be also somehow mirrored in clinical effects? So we looked at in a meta-analysis of studies that were included Chinese and studies that included white people. And it may be, although this is a very weak part, because the studies, there could be many different factors that affect the effect size. But we found that the effect size seems to be higher in Han Chinese, of antidepressant reduction. So if you really want to stimulate a Han Chinese, relative to the intensity of a white, you would have to adjust the intensity. Instead of 133% is relative to the intensity of a white that receives 120. Anyway, let's move on. Let's go back to TMS fNIRs, because this is the topic. All these moderators of stimulation-induced hemodynamic changes we wanted to replicate. So we did replicate a few, age and sex, for example. We also found that education seems to have an effect. But what we really also want to know is whether this can be somehow validated with fMRI, our HBO and HBR changes during the stimulation. And this is the reason why we perform these triple things: TMS, fMRI, fNIRs in the scanner. And we are collecting data now, so I cannot show you anything for now. Let's go back. The advantage of TMS fNIRs is that we can have a continuous assessment. And now my question was, always with the idea of a clinical application, could we use this signal as a prognostic tool? Could this signal tell us something about the patient, whether the patient relapses or whether the patient will respond to a future treatment? So at first we looked into how good neuroimaging can predict MDD prognosis, for example, in general, and there seems to be a fairly good predictive power across different imaging modalities. But in our case, we thought this TMS in combination with imaging as a supraphysiological challenge, similar to other challenge tests in medicine, such as the heart challenge test, cardiac stress test. So could the stimulation induced change, could this give us more information about the function of the brain, or the presumed abnormality of the brain in patients? Or could this tell us something about predict something, for example, whether a patient will respond to a treatment or not? One study that was just accepted finally, in which we showed that the fNIRS signal can kind of predict a function of the DLPFC, which is one of the core functions, working memory. But let's continue. The more important thing for a psychiatrist was, can we predict treatment response? And so we performed this concurrent TMS fNIRS before patients received 4 weeks of rTMS ITBS treatment. And then we looked at the response and remission rates, and we asked, can we predict the response and remission based on by our fNIRS signal, TMS-induced fNIRS perturbation. And again, what we find is during the stimulation, HBR goes up, HBO goes down. And we found that actually we can predict treatment response with our signal in combination with also clinical factors. But these imaging factors were also relevant. This is submitted. I hope you can read the paper sooner or later. We also found that the HBO change during the stimulation is correlated with the clinical response after treatment. How much time do I have left? Yeah, I'm almost done. And now this is another study that is ongoing. The question, can we also predict something else? Can we predict if a patient who had a depression before, who is currently remitted, will have a depressive episode again in the future? Because as you know, depression, or as you might know, depression is highly recurrent. If you have one episode, you are likely 50-50 roughly to have another episode. And if you have 2 episodes, it's even more likely to have a third one, and so on. If you have 3 episodes, it's 90%, something like that, so that you will have another episode in the future. But 50% of the patients do not have a second episode. So we wanted to know, can we predict whether a patient will have or not a second episode or a third episode? So we performed this longitudinal study, TMS fNIRS at the beginning, and then in remitted patients. So they had some residual symptoms, but they were all remitted. And then we follow, and currently still follow them up over 2 years in total, and ask them whether they have a relapse or not. And in the end, we want to know whether our signal can predict that. What we did for now is we compared patients who are currently depressed with remitted patients and healthy controls, and find that during as well as after the stimulation, there is already a nice difference. And we also asked, we checked whether the signal is correlated or can predict or tells us something about the previous episodes. And this is this. Yeah. And it seems to be the case, yeah, that the number of previous episodes seem to be reflected in the signal right after ITBS. Yes, and this is the summary, and I'm good in time. So TMS is an effective brain-based therapy for treatment in psychiatric disorders, especially depression. TMS effects can be assessed online utilizing concurrent neuroimaging. Concurrent TMS fNIRS has several advantages over other neuroimaging tools, such as less susceptibility to TMS-induced electromagnetic interference and cost effectiveness. And the concurrent use is a potential prognostic tool to predict treatment response in disease relapse. Thank you so much for listening. This is my group, and I'm at the end.
Thank you
I
so
will stop
much,
there.
Georg. It was an amazing presentation. It is very exciting. It's such an important work that you're doing, so many different projects. And I'm really looking forward for your fNIRS fMRI results. The concurrent TMS fNIRS fMRI would be very interesting. The translation of both TMS fNIRS, TMS fMRI. I think that would be very important for the field for the whole brain stimulation. And then we have a few minutes for the questions, but we don't have a hard cut. So if someone needs to leave in 2 minutes, this webinar is recorded and we can just continue. If you have any questions, please feel free to ask them in the chat. And we also have one question during the registration, and it was how to reduce the impact of head motion on fNIRS during high intensity TMS.
Yeah, so for the ISS system, not the Borealis, for the other system that we use, we have a little bridge. I'm not sure if you saw that. A bridge that separates the TMS coil from the optode. And for the Borealis, we fixate the head with the cushions, cushions and so on. And then we also have a sheet of paper or a block of paper that we put in between, and then if everything is set, we remove the paper, so the TMS coil doesn't touch directly the optode. As I said before, the TMS can be painful, and patients move, especially in the first 2 seconds of the ITBS. You start, you have a startle, huh? But after that there is no much movement, especially if you really fixate the head. That would be my response.
All right. So do we have any other questions? We have just one comment from Professor Sato that it's very good presentation. Thank you. And now we have another question. It was mentioned that it's possible to compute the electrical field distribution arising from the TMS. Does it make sense to use this field distribution to optimize the position of the optodes for fNIRS?
Yeah, that is a very good idea. We haven't looked into this yet, but thank you for this idea. Yeah, why not?
Yes, I think that's very just to place the optodes based on your field distribution or the TMS. And how do you think, it's just one question for me, how big is the area from the TMS pulse? So how much should we cover with our optodes to be able to capture the TMS response properly?
Well, the regions, the brain is highly interconnected, so you would have an E field that is quite large, and sometimes not necessarily directly beneath the coil, depending on the gyrification, individual anatomy. But you may also be interested in areas that are not directly below the coil, because who knows, maybe it is the clinical relevant activity that is induced by the or modulation that is induced by the stimulation, maybe somewhere else by a connected region, maybe just a little bit next to the coil, because we don't even know yet what is the optimal intensity. So maybe the optimal intensity is a bit lower than what we stimulate, and that means the optimal intensity is applied to a region that is slightly next to the coil or below the coil. So there is so many things we don't know. There's of course many things we do know, but I don't know. We just need to figure that out, how much we have to cover in order to answer our questions, whatever they are.
Yes, absolutely. Thank you. We have one more question. Considering MEP, we can see how hard it is to find the right spot to be stimulated. In this sense, how can we locate the spot in DLPFC?
Yeah.
So I think it's about targeting, in DLPFC.
So there is many studies that look into optimization of targeting, and the standard protocol was to go 5 centimeters from the motor cortex to the front in order to stimulate the DLPFC. This, of course, is very suboptimal, because different head sizes will stimulate at different regions. So then the new idea was, instead of 5 centimeters, let's go 6 centimeters, 7 centimeters, but this still doesn't account for head size. Then the new idea is we use electrode positions, and then people realized if we, F3 for example, if we stimulate slightly a bit anterior and lateral, we would be even better. So there is a new algorithm, beam F3, called that is now most optimal for standard rTMS treatments where you don't have a MRI of the patient or functional connectivity analysis. There's other studies that showed that one specific connectivity assessed with functional MRI, resting state functional MRI, seems to be relevant for the antidepressant effect, and that is an anticorrelation to the subgenural ACC, subgenural anterior cingulate cortex. So the region at the DLPFC that is most strongly anticorrelated to the subgenural ACC seems to be the best to stimulate. And this has been replicated many times. But then newer studies last year published showed that this accounts actually only for a very low percentage of the treatment response. So again, in doubt whether this is really relevant. But there is a lot of things going on in terms of the target optimization. And some people do online estimation of the E field and then stimulate at the DLPFC, anatomy based. So they do not use the MEP on the motor cortex. Yeah, this would be my response.
Yeah, thank you. And any other questions?
I have a similar question on the targeting. DLPFC stimulation can, through the parasympathetic mechanisms, can slow down the heart rate. Could you see something in the fNIRS signal? Could you derive the heart rate from the fNIRS signal and use that as a measure of that you're hitting the right DLPFC?
Yeah, that's also a very good question. And we haven't done that. We haven't looked into that yet. But Martin Arns is doing that, right? This heart rate guided navigation. And he uses EEG and he uses fMRI. But we haven't looked into the fNIRS signal yet. There's just so much you can do. Thank you.
All right, thank you so much. I think with that we can finish today's session, and it was amazing. Thank you so much, Georg.
Thank you. Have a nice day,
Bye.
evening, morning. Bye-bye.
Bye.